Hello, my name is Bradley Alicea, and I'm going to talk to you today about critical periods as developmental neurosimulation. So we're going to talk about critical periods, which are a window of developmental time that features an enhanced ability to learn. From the diagram on the bottom, you can see that these windows increase plasticity in the organism, and you can have multiple critical periods across development. They can either be overlapping or distinct. And today we're going to talk about these in computational agents. We can talk about critical periods versus development in two ways. The first is that we have this developmental trajectory. And in our case, what we're going to do is we're going to assume that no acquisitional plasticity occurs outside of the critical period. So these windows are going to be the only time that the agent can learn anything. The second is that in terms of the critical period itself, plasticity is defined by what we'll call the plasticity encoding. And so the plasticity encoding can be seen here. But first we need to have a developmental switch. So we need to turn the critical period on and off. So when the developmental switch is at state 1, it's on. When it's at state 0, it's off. Next we have the plasticity encoding itself on the bottom, which is a series of genetic algorithm genes that are expressed when the developmental switch is turned on. So these are instructions for innate features, and they interact with the environmental information that can be seen up at the top. This is an agent inspecting things in the environment. And we use a concept called Gibsonian information to characterize this information in the environment. Basically, it's a Poisson-distributed distribution of things that the agent encounters in space and time. But we can take this plasticity encoding and treat it as an interaction with the Gibsonian information. It's either multiplicative or additive. And from that, we have a rate of acquisition. There are two types of acquisition as well. So during a critical period, we can have two types of acquisition. We can have behavioral acquisition, which is acquisition of information that results in behaviors. Or we can have morphogenetic acquisition, which is the acquisition of a phenotype or embodiment through developmental processes. So here we can see morphological acquisition at work. We can see that we have a single germ cell that can become a 3-layered embryo. Those 3 layers can differentiate into a nervous system, an outer shell, and sensors and effectors. And then that can become something that looks like a Bratenberg vehicle. We can also get more fancy with this, so we can have bodies that are segmented and modular and composite. So we can use this to design robots, or we could design agents with a very complex phenotype. And this phenotype is important for behavioral acquisition because it serves as the embodied aspect of what the agent is doing in terms of its behavior and its previous acquisitions. So we can actually even draw a phylogeny here where we can go from the zygote to the 3 germ layers, and then we can classify these agents in terms of acquiring segmentation or acquiring axial elaboration. So we can grow these in different ways and have whole lineages of agents. And so this is the additive case for behavioral acquisition. We have an external communication channel where all of the environmental information is encoded. We have an internal communication channel where that is Poisson distributed, and then there's a decoding, and then behavioral information is acquired. We also have that as the multiplicative case, but in this case, it's multiplicative instead of additive. Basically, with Gibsonian information, we have a highly patterned input, a noise-free internal representation with temporal accumulation, tau, and that leads to expected patterns in the external communication channel and a minimal noisy internal communication channel. So there are a couple of examples of how you have behavioral acquisition, how the body helps to shape behavioral acquisition during critical periods. The first is lateral or directional bias in phototaxis. So depending on the source of the light here, it affects behavior. So if the agent is acquiring its light from the right side exclusively, and it has sensors on the right side, those sensors are favored for further development, for acquiring a set of behaviors that allow it to turn right in a very nuanced manner. The same thing happens on the left. If we have a left-handed bias, we can observe behaviors that are acquired that allow it to make left-hand turns in a more complicated manner. So basically, this is what the critical periods look like. We have two critical period windows that overlap, and one that's distinct, the red one. And some of these, the behavioral acquisitions and the morphological acquisitions are mixed, but as we'll talk about later, there is this contingency involved. And so when there's no critical period window, remember there's a period of no acquisition, and you can also have periods of overlap between behavioral and morphogenetic critical periods, shown between the green and the blue windows. So here we see an example of how acquisitions work in terms of developmental critical periods. So we have the rate of acquisition, and we have the developmental trajectory. So we can see that pre-critical period, the rate of acquisition is noisy but low. We've introduced some noise across the developmental period. So during the critical period, the signal was noisy and elevated in terms of what's being acquired. And then after the critical period, we see both an increase in the amount of noise and an elevated rate of acquisition. So we expect there to be a gain, long-term gain in acquisition from these critical period interventions. And we actually expect two types of interactions. One is that you might expect to see some stochastic resonance where the noisy signal will interact with the information that's being acquired and improve performance in some way. It could also decrease performance, but we don't know the full extent of that. The other thing is that there's an overlap in this case between the morphogenetic critical period, and which is not shown, and the behavioral critical period. And that interaction involves gaining morphology as you have heightened acquisition behaviorally, and so that can have some effects as well. Then we turn to using a connectome to understand acquisition a bit more, and some of these interactions between the morphological and the behavioral acquisition. And so we have this approach called the community activation encoding. Basically, we're using a pairwise matrix to characterize the connectome, and then we're going to look at this over time and look at each synapse or each pairwise connection to understand this critical period. So we can take this pairwise matrix, we can take it at each point in time, it's a discrete set of points, and we can treat each point in time as a determinant. So you can turn a matrix into a determinant and then compare these determinants over time. The reason we do this is because this is a growing network, so the number of interactions, the number of ij in this matrix vary widely over time, so we want to use something that's standardized. And we can map this to a small connectome. This is one of our composite agents with a composite phenotype. It has different features in it, wheels and sensors and different parts of the body. And these transparent circles are, of course, the connections, the sites that we're looking at in terms of the connectome. And so we have this, it's irregular, I guess, in the sense that it's not square or it's not circular. So we can actually look at this, and each one of these wij points are going to represent some aspect of this rate of acquisition and interactions between environmental information and this plasticity encoding. So we can see from this graph, we have 4 elements in our plasticity encoding, and for each element in that plasticity encoding, we've mapped out a function. And so we can see that so the red and blue are the 2 plasticity encodings that have a higher value. They have a value between 0 and 1, so the values above 0.5 are in the red and blue, and the ones below 0.5 are in the green and black. So you can see that they vary over time. This is for an extended critical period where there's acquisition going on. You can see that there are these spikes in acquisition over time. And then you can map this to the connectome, so you can get regular connectomes. You can get connectomes that are sort of on the edge of chaos in D, and then you can get these randomized connectomes in C. So finally, we can build a contingency map. So the idea here is that certain morphogenetic things have to be in place before certain behavioral acquisitions and vice versa. So the eyes need to be there before you can acquire light. And so you can see we have this map of behavioral acquisitions, and which are contingent on certain morphogenetic acquisitions being there. And we have our duration and amplitude of the critical period, which can be formulated as a way to sort of vary what kinds of morphogenetic acquisitions and their intensity with respect to the behavioral intensity and vice versa. So thank you for your attention. we have a preprint on the Sci archive